Tēnā koutou katoa, nau mai, haere mai, ki te Aotearoa New Zealand Food Safety Science and Research Centre. Ko Vivian Hunt ho, ka nui te mihi nui, kia koutou katoa. My name is Vivian Hunt, and on behalf of the New Zealand Food Safety Science and Research Centre, I'd like to welcome you all to our monthly spotlight on food safety science. So today we're really pleased to welcome two of our centres, or two of our PhD students who are associated with the centre, who will share some of their groundbreaking insights that are applied to two case studies, lettuce and seafood. So first up, we have Emmanuel Kayer, who's a PhD student in the Department of Food Microbiology at Massey University. Emmanuel will present findings that provide new information on the colonisation of lettuce with Listeria, and he will also talk about a novel method to control Listeria contamination in lettuce. The next student is Laura Bessie, who's a PhD student working at Cawthron Institute on research focused on understanding the source of tetrodotoxin in New Zealand clams. So I'll just briefly run over the bios of both these great students. Really pleased to be highlighting their work today. So Emmanuel is, as I said, a PhD student at the Department of Food Microbiology at Massey University in Palmerston. His research work involves control strategies for fresh produce. His current research is using the use of UV stress to reduce Listeria monocytogenes survival and its biofilm formation on lettuce surfaces. Emmanuel has worked on several projects in different laboratories, which include the Food Microbiology Laboratory in the Middle East Technical University in Turkey, Massey University Food Microbiology, Palmerston North, and until recently, he was an environmental technologist at the mill of OG Fibre Solutions, where he's involved in qualitative monitoring of the environment to ensure its safety. Manuel's going to submit his PhD hopefully in October, so we're wishing him lots of luck for that. So Laura is a PhD student at the University of Waikato, and she has her Master's in Science in Environmental Toxicology from the University of Joseph Fourier in France. She's been a senior research technician with Coastal and Freshwater Group at the Cawthron Institute in Nelson since 2014. Where she has been able to gain a lot of experience on the study of harmful algal blooms, and her expertise has been built using molecular techniques to research seafood safety and toxic microalgae. This has been part of an MBE-funded Safe New Zealand Seafood program, and in 2017, Laura received a scholarship from the centre to start her PhD to investigate the source and transmission of this tetrodotoxin, I don't know if I'm pronouncing that right, pufferfish neurotoxin in New Zealand bivalve. So she's hoping to submit her PhD at the Waikato University Engineering Science Department at the end of this year. So what we'll do is we'll get Emmanuel to speak first, and then we'll have a few questions after that while Laura switches over, and then she'll present her findings, and then we'll have more questions. So please look for lots of questions for these great students. So thank you, everyone. I hand over to you, Emmanuel.
Thank you very much, Vivian. Thank you for the nice introduction. Hello, everyone. Welcome to my presentation. So my PhD research work is about how to control Listeria monocytogenes on fresh produce surfaces. And for me, my research is mainly involving lettuce. So we know Listeria monocytogenes, gram-positive bacterium, which has a high mortality rate compared to other foodborne pathogens like E. coli and Salmonella. And in recent years, the issue of Listeria monocytogenes on fresh produce has been regularly reported all over the world, in America, in the States, in Europe, in Australia. With fresh produce, people are always complaining about Listeria, where we have a lot of recalls and food safety issues associated with Listeria. So, just not in the world, in New Zealand too, we have issues with Listeria monocytogenes associated with our fresh produce. And just if you go to the MPI website, you can see a lot. Just this year, in 2020, we've got about 3 food recalls of fresh produce, which is associated with Listeria monocytogenes. So this was in July 25th this year. This one was in March 20, this year, and this one was on February 8, 2020, this year. So you can see the prevalence and how regular, the consistency we are having with our fresh produce industry with listeria. And I'm very sure when there is a recall, it's going to generate a lot of economic loss or financial loss or revenue for the industries. Not just this year, in the previous years, like in March in 2017, 2016, there has still been a bit of listeria contamination on fresh produce and recalls all over in our retail and at retailers in supermarket. So due to what we have been seeing or the trend, we did a survey. We recently conducted a survey of listeria monocytogenes in bag and on-bag lettuces in Palmerston North. So we used 2 main supermarkets, and we found that bag lettuces, 7% of bag lettuces have listeria monocytogenes. There was no contamination in the on-bag lettuces, and we realized the bag lettuces they were having a high relative humidity, and we know moisture exacerbates the growth of listeria monocytogenes, and we were having about 2 to 2.5 log. So this proves the point, or this supports the point where we have been seeing a lot of foodborne recalls with listeria in our fresh produce industry. So with the control, my research work, I'm looking at using hydroponic grown lettuce and soil grown lettuce. So the hydroponic ones, because they are grown in a controlled environment, we assume microbial colonization might be lower. But for the soil grown lettuce, the soil contains many microorganisms. Some can be opportunistic pathogens. So the assumption is we will have a less contamination in the hydroponic grown one. So this is what we compared. So we first look at the differences between the two in terms of the microbial community composition. And we found out that the soil grown lettuce, majority of them had bacillus. And for the hydroponic grown lettuce, majority of them had pseudomonas. Now, the hydroponic grown lettuce had a higher microbial diversity. From the literature available says if a species or a plant has a higher microbial diversity, in one way or the other, it's able to reduce listeria contamination, because listeria has to compete for space and nutrients and other stuff. Now, from the differences, we look at attachment, how listeria attaches to the surfaces of both hydroponic and soil grown lettuces. And we realize there is no significant difference. Even under minimal exposure times of 1 minute or 2 minutes, when listeria gets the chance, it's going to attach strongly to the surface of our lettuce leaves. Not just attachment, after attachment, it's able to survive. And we did a research where we look at lettuce juice from hydroponic and soil grown lettuce. And our research, our investigations revealed that the lettuce juice support the biofilm formation of listeria monocytogenes on stainless steel. Now, in the fresh produce processing industry, the washing, the cutting, and all those stuff is being done on stainless steel. So this is listeria forming biofilms on stainless steel coupons. So this might be one of the reasons why there is recurring of contamination. So if there is listeria biofilms on your stainless steel coupon in the processing industry, if you don't really give it a very good wash, there can be a recur of contamination. As a control method, my control method is based on UV stress. Now, most of the literature available, they look at direct UV application, where you've got your bacteria on the leaf surface and you use UV as a direct kill. However, I'm using the other way around. So I first get my lettuce plant, then I put them under UV stress. I stress them just at 1.3 kilojoules per mole. This is a mild stress, because if you use a higher dosage, it's going to damage your UV leaf. It's going to damage your lettuce leaf. So you stress them, then you wait before I inoculate with listeria, then I do the enumeration. This is what happens when you don't use the appropriate dosage, like 2.6. It's going to burn your lettuce leaves, and it's not going to be good in terms of sensory, the qualities of the leaf. So after UV stressing, in terms of regards to attachment, you can see the UV stress lettuce have a low contamination, or low incidence of listeria, if you can see on here. The same thing with survival and growth, the UV stress lettuce was reduced in terms of listeria growth and survival. But the ones which were with the high dosage or inappropriate dosage, you can see there was a support. It supports the growth of listeria monocytogenes. So what's the mechanism behind this? after UV stressing, the plants contain polyphenols or phytochemicals. When you UV stress them and you wait for 24 hours, these polyphenols accumulate. And some of these polyphenols have been found to be antibacterial, or they're able to inhibit bacterial growth. For example, in lettuce leaf, we have caffeic acid, we have gallic acid, ascorbic acid. So after stressing, these polyphenols do accumulate, and these are able to prevent the growth of Listeria monocytogenes. So in conclusion, both hydroponic and soil-grown lettuce do support the attachment, growth, and biofilm formation of Listeria. And in the processing environment, the industry should really be careful about the biofilms being formed on the stainless steel or other processing surfaces, because lettuce juice supports the biofilm formation of Listeria. And mild UV stress with the appropriate dosage can significantly reduce the attachment, survival, and biofilm formation of Listeria. In terms of future work, it will be very interesting to know, in terms of bag lettuce, the relative humidity, in terms of modified artificial packaging, how they do it, so that we can maintain a very constant environment in the bag lettuce, which can help to reduce Listeria growth, at the same time keeping the lettuce in a good state. Also, the microbiota or the bacterial community composition of hydroponic grown leaves should be explored further. And also, the use of mild UV stress can be further explored in terms of gene expression, what happens after stressing the genes, the changes in the gene, the antibacterial effect, the phytochemicals, and because there is a high potential that can be used in the industry to reduce bacteria growth and Listeria growth. I want to say a special thank you to my supervisors, Professor Steve Flynn, John Palmer, Graham Fletcher, and Jason Wargent. And these are my references. Thank you very much.
Thank you, Emmanuel. That's great. So if there's any questions, please send them via chat. But one question I had was your use of the UV stress. I mean, how practical will that be for our industry to use?
Yes. So you can see in, like, one of my supervisors, Jason Wargent, you can see biolumic. They have plants being grown under UV. So this can be explored, like you get your, maybe before the plants do enter the processing environment, or before they enter, because it's able to reduce the spread of Listeria by increasing the accumulation of phytochemicals. So it doesn't have to be a big UV stand, but along the processing line, maybe there can be some stress involved, maybe passing through and continuing. I mean, when the plants are being processed or along the line. So there's a potential to be used in the industry. Okay.
Oh, thank you. We have a question from Catherine. Is the lower rate of Listeria contamination in the non-bagged lettuce solely attributable to the lack of growth, or is it a difference in industry practice?
Well, during our survey, we realized that when the on-bag lettuce, which they bring to the supermarkets, they do get a very low shelf life. I mean, after maybe 2 days, they take them away and bring in new ones. So meaning, even if there is a contamination, Listeria doesn't have the time to grow. But the ones which are in bags, they keep them there until their best before date, maybe. So they take them, they remove them just before their best before date. So, and because they are in bag and they are able to spend enough time on the shelves, it gives Listeria more chance to grow. Yeah.
So the moisture levels in the bags have quite a part to play.
Yes, it does. It's really the relative humidity of the moisture really exacerbates the growth of Listeria.
Okay. Another question: have you seen differences have you seen differences in contamination with different types of lettuce, smooth surface versus curly leaf varieties? If so, are there differences in the effect of the UV?
So for my lettuce, I only use butter crunch lettuce, but the literature available do suggest that, for example, leaf surface roughness do affect the potential of UV light, UV stress on lettuce leaf. For example, if you have a leaf with rough surface and you stress the rough surface, not all the leaf surface can be exposed to the UV light. But if there's a smooth surface leaf, all the leaf surface can be exposed to UV light, and most of the phytochemicals, every part of the leaf can be very well protected against the Listeria growth. So there is leaf surface or leaf surface, the roughness of the leaf, other things like hydrophobicity, they do affect the efficiency of UV stress and UV light on leaf surface.
Okay, well, thank you. And another question from Stefan is, did you use bagged whole lettuces or chopped lettuce salads in bags?
Chopped lettuce salads in bags.
Okay.
Chopped lettuce, yeah. And that's one thing. The chopping, with the chopping too helps the moisture. Or it because you are cutting, it helps to release a bit of moisture. So which can also be part maybe attributed to some of the moisture content in the leaf because you are cutting.
Okay, I'll take one last question and then I'll get you to switch slides with Laura. Do the Listeria monocytogenes penetrate the lettuce tissues?
Well, I am always looking at the surface growth. However, there have been a lot of reports about internalization of E. coli, Salmonella, and Listeria in leaf plants. But still, research is going on. But there have been reports about them. But for mine, I just inoculate it on the leaf surface and see how it goes on. Yeah.
Okay. Well, that's great. Okay, do you want to just switch, stop sharing your screen now, Emmanuel, and then we can have further questions for Emmanuel later, but we'll let Laura present now, and then we can have questions for both of them. Thank you.
Thank you.
Thank you. Laura, is
it working now?
Yeah, it is. It's great.
Cool. So hi everyone. So thank you, Vivienne, for the introduction earlier on as well. So my PhD project is looking at the source and transmission of tetrodotoxin in New Zealand shellfish. So first of all, what is tetrodotoxin? So I'm going to call it TTX for the rest of the talk. It's a potoneurotoxin. So 1 to 2 milligram will be enough to kill a human, and it does so by blocking the sodium channels. TTX was first detected in pufferfish, but since there's a lot of research, it has been detected in a lot of genetically different organisms, so in marine, freshwater, and terrestrial organisms. So here you have a few examples of the organisms that have been shown to have tetrodotoxin. So the blue ring octopus, some flatworms, starfish, bacteria, some worms, some terrestrial toads as well, and newts. What is really interesting about TTX is that the source and the biosynthetic pathways are unknown. So no one really knows where the toxin is coming from and how all these different organisms are actually the toxin. So there's 2 or 3 main hypotheses regarding the origin of tetrodotoxin. So the first one is that the toxin is produced endogenously. So the organisms are producing it themselves. Newts have been shown to retain high amount and even have higher amount of tetrodotoxin levels when they're kept in captivity over several years. On the opposite, a lot of research has been done showing that the toxin is exogenous, so coming from the external environment, mostly from the diet. These slugs, on the opposite to newts, have been shown to depurate really quickly the toxin when they are fed a non-toxic diet in the lab. And the third hypothesis is that something from the diet, but when they're entering the other organism together, they are producing the symbionts that produce the toxin. So concerning the origin, bacteria is definitely the most widely accepted hypothesis, and the research over the years has been over 150 TTX producing strains. Most of them are Vibrio and Bacillus. There's a problem though, it's not always the case. So for example, the newts that have really high amount of toxin, they haven't found any bacteria that could be responsible for the production of tetrodotoxin. Also, all the bacteria that have been shown to produce the toxins, they really produce a very small amount that wouldn't really account for really high concentrations in the high organisms like pufferfish or the slugs. And also, most of these studies on the bacteria producing strains, with all the chemical method methods, and it's not really robust. So there's probably been some contamination, or also it was a lot of false positive. So a good question that we had, because my background is a bit in microalgae. So why couldn't microalgae produce the tetrodotoxin? So a lot of people would know that dinoflagellate, like Alexandrium, produce the harmful algae bloom. They have the mussel industry closing the farms. Cyanobacteria produce a lot of harmful algae bloom in freshwater environments, and they produce a wide range of toxins. So and no one has looked really into algae as the producer of tetrodotoxin. Just quickly, a little bit of background. Tetrodotoxin was first found in New Zealand in 2009 when a lot of dogs died in Auckland beaches after eating and licking the sea slugs that you have at the top. And the slugs were found to have a huge amount of toxin, so 1.5 grams per kilo, which is highly toxic. And after that, a lot of research was done in New Zealand, and it was found in flatworms and also in shellfish. So PPs were found to have 0.8 of a gram, and also in some moist mussels and some oysters. So it was quite interesting because in 2017, the European Food Safety Authority released a recommended label that the TTX should be under in shellfish in Europe, and at 0.04 milligrams per kilo. That's the level where you will not see any effects on human health. It was quite interesting because, you know, we have shellfish in New Zealand being above this, and even though it's not really a risk, human just yet, because you would need to eat a huge amount, it was quite interesting to know where it would be coming from. So I decided to look at PPs. So they're a really good study organism because, on the contrary of slugs that move around and they're probably eating each other, PPs are good because they're stationary and they filter feed. So if they do accumulate the toxin from an external diet, then wherever they are, the toxin distribution will be there. Also, they're an important food source in New Zealand and really abundant. So we thought they would be really good to look at that. So I had, my PhD was divided into 4 objectives. So I was looking at the distribution of tetrodotoxin in different PPs population around the country, to look where the toxin is internally distributed, so which organ accumulate the toxin. That would tell us a bit more if they come from an external or if they are producing the toxin themselves. Do PPs depurate or accumulate the toxin when they're kept in captivity? And then, after all these questions I answer, looking at the primary source using molecular techniques. So I had PPs from different sites in New Zealand. So the sites are ranked from the top of the north to the bottom of the south. The Hokinga Harbour is the site that has the highest amount of toxin, quite significantly. And you can see that the PPs in the South Island barely have any toxin. It was a significant difference between the North and the South Island. It was really interesting. So they tell us that the production of tetrodotoxin could be triggered by warmer waters, which what has been found in Europe as well. The warmer sites also tell us that it could be a bacteria producing it, but also cyanobacteria or dinoflagellates get triggered with warmer waters as well. And is the producer mainly in the North Island? So maybe there's something that hasn't really crossed the Cook Strait or something. We're not sure, but I will be looking also in genetic differences between the PPs and see the production or would they have some way of accumulating it. So then I looked at the macro distribution. So I divided PPs from the Hokinga Harbour because it's a bit of a hotspot, and we knew before I started my PhD that the toxin levels stay quite high for the whole year. So I collected PPs and did some dissection. So I split them into 5 organ groups. And the siphon where the organs accumulating most of the toxin. So that was really interesting because that's the organ that it's in contact with the most out of the water. And also it could be as a mode of protection because you know, shouldn't be grazing on siphon that are toxic, which has been shown for clams in the Pacific that accumulate sexy-toxin, which is another neurotoxin. So that was quite interesting. And I looked, I used immunohistochemistry, the tetrodotoxin into different organs. If you can see my mouth, but all of the more darker brown color cells is cells that have accumulated or binded the toxin. So here, that's a loop from the outer siphon. You also have it all around here. So it was really interesting to confirm the chemistry methods. It was also found in the labial pulps and the gills, which is what is used for the pipis to filter the water and also to sort the food. So again, it's another organ that's in contact with the external water and used in the diet. And lastly, it was found, and so here there's the crystalline sac from the digestive gland for the pipis. So again, the toxin was all on the outer layer as well, so direct contact with the water that they have filtered. And also it was in the digestive tract in the stomach. So again, that tells us that the toxin is likely coming from an external external source in pipis, because it wasn't found also in the gonads, so they can be really transmitted to the second generation of pipis. So that wasn't really a trait that was. So after that, I wanted to look. So I thought that pipi would be depurating the toxin over time. So I collected 200 pipis from the Hokianga Harbour and kept them in the lab for over 150 days. And we could see that they clearly and significantly depurated the toxin. So that was telling us that they do take the toxin from an external source and stop producing it when they're not in contact with whatever is in the water there. And I also dissected them again and looked what happened in the different organs over time as well. So you can see that the siphon, again, had the highest amount and decreased significantly over time as well. So they could be binding the toxin into the siphon, and then after time not being exposed of the toxin producer, they will be likely losing it. And what was interesting as well, it's in the digestive gland, there was quite a bit of toxin. It completely went away after 20 days. So again, bringing to the hypothesis that the source of the toxin is likely an external one. So I'm currently finishing my last chapter, looking at if PP can accumulate the toxin from just being fed. So it would just find a method to encapsulate the toxin, because the tetrodotoxin is water soluble, so we couldn't just feed it to the shellfish and accumulate them. So, I made some agar and gelatin capsules with humic acid. So humic acid is what is present in soil and also can bind that water-soluble toxin. And then we fed those capsules with algae to the pipis. And I'm looking if they do accumulate it. And I think they have. So it's been really interesting. And it's a good point for future studies where we can feed it high level to some shellfish and also feed the shellfish to, lobsters or fish and see how the toxin will get accumulated. So if tetrodotoxin stays or concentration increase in New Zealand, that will be really important to know for the industry and see how the organism that feed the shellfish will behave. And so I looked from the PPs from the first study that I looked at all of the different concentration from the different population. I wanted to have a look at potential producers. So I've dissected all these PPs into the siphon and the digestive gland, which were the two organs that contain the highest amount of the toxin. And I looked at the different bacteria population and communities that were inside them. So they are quite different from one side to the another, which would make sense because temperature are what drive bacterial families in the water. So it makes a lot of sense that they are different, from the top of the North to the bottom of the South Island. But what was interesting that you can see in what light yellow in the Hokianga Harbour in the digestive gland cyanobacteria, and also in Mahia, which were the two that had the highest amount of toxins. So that's really interesting. So I dived a bit deeper. And what I did, I took I took the sites that didn't have a high amount of toxins. So it was Marahau, Riverton, and Dunedin. So I pulled together all of the bacterial community, and I subtracted those to the Hokianga Harbour sites. So the way of thinking was that whatever was producing the toxin was in Hokianga, but wasn't in the low, abundance in the bottom of the South Island. So I looked whatever was left that would be potentially the toxin producer. And so we found that Bacillus, Synobium, Pleurocepsa, Pleurococcus, and Vibrio were present in the sites with the high amount of toxin, but were not present in the sites with low amount of toxin. Vibrio is the one that's produced tetrodotoxin, so it was interesting to see it there, and we could try and culture it to see if they do produce a toxin. A lot of labs around the world are already doing this, so we thought we should focus on the other ones. Synobium, Pleurocepsa, and Pleurococcus are 3 picocyanobacteria, so they are very small cyanobacteria that are present in the oceans. And some of the freshwater picocyanobacteria have been shown to produce toxins, freshwater toxins. So it's really interesting to find that cyanobacteria were present, but in the site they had the toxin, and could be a really good study organism to look at the toxin production. So we definitely will be looking at culturing them and looking at the toxin production. I also look at the core microbiome of the pipis for the Hokianga Harbour. So they were collected every month for a whole year. And I also looked at the bacteria that were present 70 to 90% of the time in all the sites over the whole year. And you can see again, so mycoplasma and spirochete at what is the digestive gland made of, so that was normal to see it there. But again, there's Synechococcus, Synobium, and Pleurococcus, which also, again, are the cyanobacteria. So it's a very interesting new lead into the production of cyanobacteria. I'll be looking at other shellfish in different countries to see if they do have cyanobacteria as well when they have tetrodotoxin. I'll also be looking probably at dinoflagellate communities and see if there's some classified dinoflagellates that could be producing the toxin as well. So just as a conclusion, so yes, PP do depurate. It's only present in the organ that are linked to the diet and to the external environment. So it's very likely that the source is endogenous and the diet source for the PPs, which could be a little bit problematic if whatever the source is increases the abundance with climate change and the warmer waters. So it's definitely worth keeping an eye on. So like I mentioned, isolation and culturing of potential producers of cyanobacteria is really high on my list. And looking at bioaccumulation for the food chain once my first method of encapsulation is developed. Looking at dinoflagellates and other eukaryotes in the digestive gland of the PP. So thank you, everyone. And if you have any questions, let me know.
Thank you very much, Laura. That's great. We do have a question there already. It's a question from someone about how can you define TTX from a biochemical point of view? Is it a protein?
No, it's actually I'm not quite sure. It's just a molecule, yeah, but it's not quite a protein.
It's not. It's okay.
Okay.
Another question from Tim is, how do the TTX levels observed in PP compare to, say, slugs, pufferfish, and blue-ringed octopus?
Oh yeah, so they are a lot lower. If I can go back. Yeah, so the blurring octopus and the slugs and the pufferfish will be all in those range of the sea slugs and above. So they're really toxic compared to the shellfish.
Oh, okay.
So yeah, I think you would need a few kilos of PPs from the Hokianga Harbour to actually get sick. But
You a lot. That's good to know.
Yeah,
I need to warn my sister.
No, yeah.
And from Catherine, she says, great talk, Laura. Was the TTX present in the hemolymph? Also, has there been any known illnesses associated with pippies from the Hokianga that might be related to the TTX?
No, so he wasn't in the hemolymph. No, there hasn't been any I'm aware of in the Hokianga Harbour. I know that sometimes they do have sexitoxin. They have some signs up to not eat the shellfish. But I'm not sure. No, not that we're aware of. There hasn't been anything.
Does anyone else on the call know that, Graham? No, okay. Well, if you've got any other questions for either Emmanuel or Laura, those are both great talks. Thank you. And I was just wondering about your talk, Laura, was so you're sort of saying that the bacteria, you noted the higher levels of Vibrio and Mycoplasma and Hokianga, so you sort of can use the bacteria as a biomarker.
Well, we're not quite sure they do produce that, so we need to
understand
it. Culture them, and then once we confirm that they do produce that, then we can look at more studies and also looking at the genes and things that could be producing the toxin. But it's been such a mystery for so many years that no one really, and no one has been able to find a producer, but no one has looked into cyanobacteria. So I think it's promising.
Okay. Any other questions from people on the call for either Emmanuel or Laura? No. Well, I think I'll just, we're sort of almost up to, is there any more questions? No, I don't think so. So thank you very much, both Emmanuel and Laura. I think those were both excellent pieces of work that you've presented, and it's great to see this work coming through and leading new studies and the future of our work. So we do have our next seminar, which is going to be held on the 23rd of October, rather than the last week of October, because we have the whole genome sequencing conference being held the 28th and the 29th. Which is being it's a 2 days of 2 hours on whole genome sequencing with speakers from all over, including some of our centre people. And so we're going to hold our next seminar on the 23rd, and that's going to feature the work of 2 more PhD students as well. So hope that you can all join in and hear about some of this groundbreaking research. So thank you very much, both Emmanuel and Laura.
Thank you.
Thanks, everyone.